Hi, my name is Virginia Krause, and I'm a fifth-year PhD candidate at UC Davis in the Agricultural and Environmental Chemistry graduate group. My academic journey started in my undergraduate institution, where I studied chemistry and environmental science. And when I was looking for graduate programs, I wanted one that merged these two programs together. So that's how I ended up working at UC Davis in the Poulin Research Group. Our work in the Poulin Lab focuses primarily on investigating the fate and transport of trace metals in the environment, and my work specifically looks into the dynamics of mercury in freshwater ecosystems. So why am I focused on mercury? Mercury is a pervasive global contaminant with a long history of pollution in California from historic mercury mines. On our map, all of the red triangles represent mercury mines that were active during the 1800s gold rush and are now releasing mercury into local watersheds, many of which are reservoirs. The problem is that this released mercury, shown in our reservoir schematic as inorganic mercury, or H2, can undergo a microbially mediated transformation to form neurotoxic methylmercury, or MEHG. This methylmercury can both bioaccumulate and biomagnify up aquatic food webs, which can cause severe health effects to individuals that consume fish. My research seeks to understand the dynamics of this transformation from inorganic mercury into methylmercury in the water before it enters the food web to better inform management strategies that reduce fish concentrations of mercury. To understand this transformation, my work addresses three major questions: where, who, and what in a specific reservoir in San Jose, California. Starting with where, we need to understand where the methylation of mercury is happening. In many reservoirs, this process happens in the sediments, but methylation may also occur in the water column. From a management perspective, if we want to reduce the concentrations of methylmercury, we need to know where to target, whether it's the sediments or the water. We also need to know who to target. As I mentioned, this reaction is microbially mediated, which means microorganisms are responsible for the methylation of mercury, but not all microorganisms. My work will identify which microorganisms maintain a specific gene sequence, the HGCAB gene, which is the only gene sequence that can methylate from mercury. So what we'll be doing is looking through the reservoir water column to identify which microorganisms maintain this HGCAB gene and whether or not they have specific characteristics like sulfate reducing capacity, methanogen, or higher redox groups like nitrate reducers and fermenters. Lastly, we need to understand what is influencing this transformation. It's not just the mercury and the microbes. Other conditions in the water column and sediments impact mercury methylation, like redox and organic matter, which can be heavily influenced by the seasonality of the reservoir. Overall, my research addresses these three main questions of where, who, and what in a mercury-impaired reservoir in San Jose, California, through 2 years of monthly monitoring, paired with 4 intensive field experiments to better understand the controls of methylation. So why does this matter? Understanding the controls of methylation will help to inform targeted remediation strategies to reduce mercury concentrations in fish within our system, which will help in the overall goal of limiting human exposure to mercury. I look forward to sharing some of my findings at the upcoming ARC Symposium, and I thank the ARC's Foundation for funding this work. Thank you for listening.